GEN-1 is a novel immunogene therapeutic designed with Celsion's proprietary TheraPlas technology. When delivered to a tumor site, GEN-1 may potentiate both innate and acquired immune responses against cancer. Advanced epithelial ovarian carcinoma and glioblastoma are lethal cancers in which the complete surgical resection of tumors is difficult to achieve and recurrences are common. Chemotherapeutic regimens have not been proven curative in recurrent disease. They do, however, induce severe toxicities. Novel immunotherapeutic approaches are being investigated in both glioblastoma and advanced epithelial ovarian carcinoma. For the purpose of this animation, we will focus on ovarian carcinoma. While traditional chemotherapy kills fast-growing cancer cells, immunotherapy enhances the local and systemic immune response against tumors, potentially offering a more targeted and possibly less toxic approach to cancer treatment. The cytokine interleukin-12, IL-12, can induce anti-cancer immunity through multiple mechanisms. Firstly, IL-12 promotes cellular production of the potent immune mediator, interferon gamma, or IFN gamma. Secondly, IL-12 stimulates the proliferation of CD8 positive T cells and natural killer cells and their cytotoxic activity against the tumor. Thirdly, it shifts the differentiation of naive CD4 positive T cells towards a Th1 phenotype, further enhancing the immune response. Fourthly, regulatory T cells that suppress immune responses may be inhibited by IL-12. Finally, IFN gamma promotes the expression of anti-angiogenic molecules, halting the growth of new blood vessels that supply oxygen to the tumor. GEN-1 consists of a human IL-12 plasmid formulated with a non-viral synthetic DNA delivery system, polyethylene glycol polyethylene imine cholesterol, PPC. The PPC polymer is condensed together with the plasmid, forming nanoparticles. The lyophilized nanoparticles are simply reconstituted with sterile water at the patient's bedside, and the solution is infused directly into the local tumor environment via a catheter. The three components of PPC play different roles in bringing plasmids to their target cells. Polyethylene imine, PEI, condenses the DNA strands into nanoparticles by electrostatic interaction. Polyethylene glycol, PEG, improves in vivo stability and protects the plasmid from clearance by serum proteins. Cholesterol is a biocompatible moiety that enhances trafficking across cell membranes. The nanoparticles can thus become endocytosed by many types of stromal cells in the tumor microenvironment. Within the cell, polyethylene imine, PEI, facilitates lysis of the endosome, allowing the naked DNA plasmid to emerge. The IL-12 gene it encodes can now be expressed by the cellular machinery. Transfected cells are able to produce sustained concentrations of IL-12 protein in the vicinity of the tumor. GEN-1 thus provides for a local and durable elevation of IL-12 at the tumor site, with exposure that persists for several days after a single treatment. The secreted IL-12 does not accumulate locally. This may minimize toxicity by avoiding spillover into the systemic circulation. Sustained concentrations of IL-12 may lead to prolonged infiltration of immune cells in the tumor environment, enhancing the local immune response against cancer. The safety, biological activity, and anti-tumor activity of GEN-1 have been demonstrated in Phase 1 clinical trials in ovarian cancer patients and in animal models of ovarian cancer. Its anti-tumoral and biological effects have also been demonstrated in a murine model of experimental glioma.